This is part 3 of a 4-part video series about the molecular biology of COVID-19 in 2022.
The mRNA vaccines work through producing mRNA that codes for the spike protein of the COVID-19 virus. The mRNA enters cells where tRNA and the resultant protein is produced using the mechanism of translation. Once a sufficient level of spike protein is produced, our immune system recognizes the protein as foreign and mounts an immune response towards the spike protein. All vaccines allow our immune system to recognize and memorize what the virus is like, where the immune response will be stronger because of this recognition.
One eternity later.
The mRNA vaccine builds memory of the spike protein in our immune system, so that when we get infected with the actual virus, the immune response from recognizing the spike protein of the virus will be much stronger, increasing our immunity towards SARS-CoV-2. You might be wondering why we would need this technology instead of the usual forms of vaccines we are familiar with. Traditional vaccines often use a weakened or attenuated version of the virus. This means that the immune system will be exposed to the actual pathogen and mount a resultant immune response. For the majority of those receiving traditional vaccines, the side effects would be minimal, if any, such as fever or runny nose. However, because the weakened virus still has the ability to replicate and proliferate within the body, serious side effects or complications could still occur, especially if the host immune system is not sufficiently strong to suppress the virus spread. This is where the mRNA vaccines differ significantly from traditional vaccines. Lily talked about scientists using gene sequencing technology to flesh out the genome for the SARS-CoV-2 virus. The mRNA vaccine takes a sequence from the genome that codes for the spike protein and transports it into our cells, so that only the protein is replicated and recognized by the immune system. Because no other part of the viral replication machinery enters our body cells, there's little to no risk of being infected with COVID-19 after an mRNA vaccine jab. However, mRNA cannot effectively enter our cells because it has a negatively charged backbone, so it cannot pass through the nonpolar lipid bilayer effectively. Hence, a vital component of the mRNA vaccines is the carrier molecule that allows the mRNA to diffuse into cells for translation. This carrier molecule is called lipid nanoparticle, or LNP for short. A fun fact is that LNP development is mainly credited to our very own Dr. Peter Cullis, professor of biochemistry at UBC. The lipid nanoparticle contains liposomes that have a positive charge to interact with a negatively charged mRNA molecule. This is then surrounded mostly by nonpolar lipids so that the mRNA can pass into the cell effectively through the nonpolar lipid bilayer of the cell membrane. The lipid nanoparticles also protect the naked mRNA molecules from RNases from breaking down mRNA, increasing its half-life within the body. Without the development and implementation of LNPs, we would not be able to transport the genetic material into the cell to initiate an immune response. A fun way to look at LNPs would be like a police escort through a crowded section of road. Cars normally cannot cut through traffic, but with a fleet of police cars flanking you, you can cut through the traffic jam in no time. LNPs are like police cars paving the way for the car and its passenger, the mRNA, to pass through the cell membrane where cars usually cannot pass through easily. Since the vaccines were designed using the alpha variant as a template, there is concern that the vaccines may not be effective against the new variants. The vaccines may not provide a level of protection sufficient for people to mount a powerful immune response to the virus. In a systematic review, the researchers looked at studies on fully vaccinated individuals. As expected, protection against the alpha variant is high at around 84% efficacy. However, the results for beta, gamma, and delta variants all show less convincing results. Studies on the beta variant show a wide range of possible efficacies from 22 to 100%, while studies on the gamma variant found that efficacies were overall lower than 50%. Regarding the delta variant, it is only found to have lower efficacy due to limited data. This shows that although the vaccines are effective against new variants, the protection offers varies greatly with each variant. There is currently not enough research done on this area to conclusively summarize the effectiveness of vaccines on emerging variants. Some scientists have proposed that a possible solution would be to modify the vaccines according to the variants, which should be a relatively straightforward step in the manufacturing process. However, this idea is still very much in its preliminary stage, where there are hopes of launching trials for variant vaccines.